So n-Lorem is a nonprofit foundation that I initiated in January of 2020. So we're a bit more than 2 years of age. And our mission is to take advantage of the technology that was created and advanced and still advancing and validated under my leadership to discover, develop, manufacture, and provide experimental ASO treatments to what I call nano-rare patients, and to do that one patient at a time, and to do all that for free, for life. I'll just say that again, for free, for life. I do think that it's time to parse patient populations a little more precisely than we have. But for the moment, let's just focus on the nano-rare, what I'm calling the nano-rare patient. This is the rarest of patient populations. Many of our patients appear to be unique. That is, based on what we know today, that patient and that patient alone has a mutation that produces a severe disease. Others are members of a very, very exclusive but not attractive club that involve maybe as many as 30 patients around the world. Since these mutations are typically de novo, those patients are spread around the world. And because of extreme difficulties in making diagnosis, most of these patients, when they're discovered, are at varying stages in these diseases. So these patients are the most isolated and severely ill, desperate patients that I've encountered. And in addition to being severely ill, typically these diseases may manifest in multiple organs. And of course, many of these mutations manifest in infancy. So about 2/3 of our patients so far are children. And obviously, in addition to the primary effects of the disease, we also have significant developmental overlays that complicate the clinical picture. And in addition to all the other challenges that these patients have, because of delays in diagnosis, most of the time when we get patients, these patients are very far progressed, and in many cases, they're in urgent need of treatment. And I'll introduce you to a patient who qualifies for that description. The one giant plus that we have in the nano-rare patient is that the vast majority of these patients have an identifiable mutation in a single gene that is causal for the disease. And once we know the mutation, whether the disease has a name or not, it really doesn't make any difference, because we have the information we need to go to work to make an ASO. So we are at n-Lorem, I think, for the first time on an industrial scale, practicing mutation-directed drug discovery and development. We are absolutely agnostic as to disease. Really doesn't matter to us. Actually, the names of diseases are mostly archaic and many centuries old and probably should be discarded anyway. We have an actionable event when we know that mutation. Now, I suspect to some of you, the idea of mounting a drug discovery and development program for a single patient and then giving the drug away for life sounds impossible. It certainly did to me, having been responsible for leading about perhaps 25 drugs that are on market, including Spinraza, the drug I think led the way in SMA. But about 5 years ago, I realized that in principle, the technology we had developed could do this. And it was such a foreign concept to me that I spent a few months just checking my numbers and checking my sanity. But as I'll show you, it can be done. This conceptual framework really is remarkable in itself, in that I think it was unimaginable before 5 years ago. Second component that I think is critically important in this endeavor is that we are industrializing the process. This is drug discovery and development, and these are complex drugs. And so every step in the process has to be of highest quality, especially the identification of the optimal ASO. And as I'll show you, we've put in processes that allow us to do that and take advantage of 3+ decades of experience. And even though each of these patients may be unique, we know now that there are millions of these patients in the world. And as more human genomes are sequenced, that number is going to grow. So any solution that you contemplate can't be just a solution for a single patient. It needs to be scalable. So industrialization of this process, I think, is critical. And we are pioneering a new model. It is a nonprofit model, and I'll walk you through why I think that's essential. But that does mean that we are trailblazing. Every step we take is a step into the unknown. And it also provides an extraordinary opportunity to learn. And I'm astonished at all the things I've learned in just these 2 years, and we're just beginning. And so we are committed to learning maximally from each patient and from the aggregate experience. We're entirely open access. We encourage our investigators to publish case reports. And as I'll show you, we plan to publish the results that we have annually and to share that knowledge broadly. And there's just an extraordinary amount to be learned here. So I guess the next question then is, what do you need to even think about doing this? Well, as Emil said, in the vast majority of developed economies, healthcare funding, despite how much it is not sufficient to provide quality care for patients with common disease. And so I think we must be aware of the economic realities of healthcare today. And for a single patient to add multiple millions of dollars just simply isn't on. We also need access to the patients, of course, but we need much more than that. We need that patient to be genotypically characterized and very, very thoroughly phenotypically characterized. We need a research physician capable of caring for that patient while administering an experimental agent. And we need institutions that are willing and able to do all that. The third leg of this is obviously a supportive regulatory environment, and I'm pleased to say we have that, and I'll come back and describe that for you. And then obviously you have to have a technology that can do this, and ASO technology is a technology that can. So why a NanoRare, a nonprofit approach? I mean, there's much to commend the commercial model. We know how to use it. It's very straightforward. It may be overly costly and overly bureaucratic, but we know how to do it. However, ASOs are supervised by the drug division, and the drug division has historically been much more rigid than the biologics division. But it's very clear there is no path to approval for these patients. It is not possible to do a controlled clinical trial or 2 controlled clinical trials to meet commercial requirements in a single patient, or even if you have 20 or 30 that are spread around the world. Probably equally importantly to me is if one were able to actually achieve commercial approval to make economic sense of it, as Emil just discussed, we'd have to charge these patients and families many, many millions of dollars a year. And I think surely we can do better than that. So one of the objectives of n-Lorem is to make it economically unjustifiable to do that. We give the drugs away. Then patient access and access to the information we need. Early on in my thinking, I was introduced to the Undiagnosed Disease Network, and I imagine most of you are very familiar with it. But you know that it was initially an NIH intramural program and then was expanded to 8 tertiary care centers and a central data hub and central facilities for gene functionalization and so on. And so that was the first collaboration I did, was to link up with the UDN. And the UDN, I think, has made very many important contributions. And among them, of course, just focusing on the undiagnosed disease patient. And we know, as a product of the work that UDN has done and our experience, that the vast majority of patients who have nanorare diseases are never diagnosed. They progress, succumb to their disease without ever knowing why their lives are shorter or less robust than the lives they see around them. So we know as we deal with the patients who are fortunate enough to make a diagnosis, that we're dealing with the tip of an enormous, enormous iceberg of misery and opportunity to learn. Interestingly, despite all the resources invested and pretty tight screening processes, to date, only about a third of the patients accepted by the UDN achieved a diagnosis. And again, I think that speaks to the need to learn more and to take every opportunity we have to drive shine a light into these spaces and learn from the light that's on them. For the lucky few who are diagnosed, average time to diagnosis, UDN data is 8 years. I would say our data so far would suggest it slightly shorter than that. Irrespective, the journey to diagnosis is long and it's perilous. Almost all of these patients will have been misreferred, misdiagnosed, and frequently mistreated. And finally, I think a key plus that the UDN can claim, and justifiably so, is that it stimulated interest. And interestingly, of the about 150 cases now that we've had applications for, less than a third have come from UDN's sites. And that speaks to the growth of personalized medicine centers around the world, which is great news for these patients. There's more people and more places to take care of them. So the UDN was a critical component in what we're doing. Next is the regulatory process. Beginning in January of 2019, I wrote the senior leaders of the drug division and was astonished to get replies from all of them within 30 minutes that were positive. Having been dealing with the FDA for almost 50 years, that was a first for me. And importantly, the FDA, at really light speed for the FDA, has completed issuing guidance for ASOs for the NanoRare patients. This guidance is highly specific to ASOs. There is no other technology that has the guidance today. And it is also specific to the nonprofit provision of these drugs. And that's truly critical. We contributed multiple proposals, and I'm pleased to say a large fraction of them were incorporated into the guidance. And we're still in discussions with the FDA. I think there are many things we can do to make these even more efficient. But this is the rule book, and it allows us to go from the gene to the patient very expeditiously with very little preclinical information. Certainly nothing compared to a commercial drug. And again, that puts a higher and higher premium on doing each step as well as possible. But that's the rule book, and it provides the rules that we and all the institutions and physicians who are working with us depend on. And we applaud the FDA for the rapidity and the quality of the response that they provided. I'm now going to focus on the technology, and I'll be making some assertions in the next few slides. I don't have time to share the data, which makes me uncomfortable, but there have been a lot of reviews of the technology of late. There's a bunch that I've written, but there are others as well. So if you want to learn more, you can easily do that. It's a long forgotten fact that when I founded Ionis in 1989, there were at least 5, I think maybe 6 companies founded by VCs to tackle the same opportunity. And of those, only Ionis succeeded, and I think there are a number of straightforward reasons to explain that. But certainly one of them is a set of decisions I made before founding Ionis, and this is germane to the NanoRare patients, so I want to walk you through it. Of course, I knew that if we were going to be successful, which seemed highly, highly unlikely, we'd have to take what was a blank piece of paper and write a textbook. And the first step in that, of course, is this is a chemically based discipline. And before 1989, there had never been a medicinal chemistry effort in oligonucleotides. And so at Ionis, we built the largest and most successful. And our program at Ionis is made and studied many thousands of different modifications and scores of different designs and so on. And that medicinal chemistry, of course, is the toolbox that's fueling modern aptamers, CRISPR, siRNA, another form of ASOs, and so on. And it has led to the major classes of ASOs that are being used clinically today. We coupled that to massively parallel screening of target sites in RNA. When we started, that was really almost impossible to do. As we look at it today, we've now screened many millions of ASOs in multiple systems and ever-increasing numbers of human-derived cells. And that has generated, I think, very sophisticated algorithms that support design of ASOs that are unique to us, and automation that's unique. And it's that combination of experience and automation that makes the discovery of an optimal ASO for us almost free. For a nanorare patient, an n-Lorem patient, we'll typically screen at least 1,000 sites in the target RNA. And we do it because we know it's necessary. If you don't, you won't get the optimal ASO. And I like to say that to do that, we just put a couple of plates on the robot at night and come in the morning and get the results. That's a bit of hyperbole, but it is an extraordinarily efficient, extraordinarily inexpensive process for us. And then all of that was coupled to the work that I led over the years and still lead to understand the molecular mechanisms by which these agents produce the phenotypes that we observe. And it's that 3+ decades of committed, integrated innovation that allows us to do what we do. It's also important for me to mention that Ionis uniquely has put together integrated safety databases that integrate all the data from non-human primates through all the controlled clinical trials for each chemical class that we're doing. And since these agents just differ by sequence, by and large, you can predict what the next drug is going to do based on the 10 that you just studied. And those databases are published. I'm the first author on the first 4, and they're available to regulatory agencies. And so armed with that, we know the dose, we have a good idea of the frequency, the route, and we have a very good sense of what we should look for in terms of adverse events, which I think is the key to facilitating doing this and doing it well. And once again, getting that optimal ASO is the critical step in this process. The technology is also much more versatile than people who aren't steeped in the technology realize. Of course, RNase H1 has been an incredibly valuable pharmacological mechanism, and we know and we understand it in detail, but we can use AGO2, you can use double strands or single strands if you have a stable phosphate that binds to the AGO2 binding pocket. And thanks to advances in understanding of RNA metabolism, we now know how to design ASOs to take advantage of a variety of other approaches that reduce the target RNA. And that's useful because on rare occasion, H1 is not the best mechanism, and so we have that versatility. More importantly, we've added a number of mechanisms now to driving alternative splicing, which we did in Spinraza for spinal muscular atrophy, that allow us to selectively increase the production of proteins. And this is all driven by advances in understanding how mRNAs downregulate their own translation. About 50% of all the messages in human cells are downregulated by sequence or structural features in the mRNA. And in fact, the first UORF ASO that will go to the clinic will probably be an n-Lorem patient. Since we discovered that mechanism, I'm very excited to see that one go into the clinic. We've also shown that we can administer these ASOs by essentially all routes of administration. And that, again, increases versatility. And in addition, we have a lot of experience in all the various organs. So there are limits. We can't fix null mutations. And certainly we look forward to the day when gene therapy can be industrialized to the place that it could tackle this. And we also choose to focus on only 5 organs, 3 routes of deliveries. Why? Because these organs we know we get great potency in these because the distribution is good. And so we have a really strong therapeutic index, and we think that's important given the limited preclinical data we have. So systemically, sub-Q, about once a month, but we can dose quarterly if we like. For liver and kidney in the central nervous system, intrathecal every 4 months or thereabouts. These drugs are nice to give as aerosol agents for lung issues. And then we have more than 3 decades of experience with intravitreal administration, which today we do every 6 months at most. Over on the right is the total annual dose for these patients. And smallest run that we can do in a GMP facility is about 10 grams. These drugs are extremely stable as dry powders. So with a single manufacturing run, we can make enough drug to supply a patient for life. And so that part of free for life turns out to be not nearly as brazen as it may sound. And manufacturing is actually a relatively small component of the cost per patient. So I do want to mention that n-Lorem has ambitions that go well beyond today. Of course, our first role is to treat the patients we can. And yes, there are patients we can't help. But I think what's important is the patients we can. That's a central dictum of therapeutics, in my view. Treat today the people you can while you're working to make better treatments for more tomorrow. I think the quality systems that I'll describe for you are critical, and we're hoping that others follow us as they consider entering this space. We know that this is a way too big a task for little old n-Lorem, and I'm very pleased with the progress that we've made in putting a network of collaborators, donors, and partners. And I think, I think in the end, it's going to take a city rather than a village, but we're on our way. We're committed to learning and we're committed to sharing. We're an open access organization, and we'd be delighted to share the data with you as we have them. I think it is obvious that as Emil said and others have said, treatment always is the tip of the spear. If you want to drive advances in diagnosis, the best thing to do is to have some treatment. Certainly that was the case with SMA. With Spinraza, we then had the opportunity to drive genetic testing for SMA into newborns. And we showed that if we treated these patients before they became symptomatic, most of these kids grew up like normal, healthy children, which is kind of hard to beat. Similarly here, I think the opportunity to treat even a fraction of the patients will drive progress. And the key step, as has been mentioned several times, is to introduce genomic sequencing into newborn screening. It's actually happening. In the UK, it's starting to happen in the UAE and in other countries. This is happening. So it will happen in America, and treatment will drive the way. And then finally, as I mentioned, these patients are isolated. They have no one to talk to. And so we're working very hard to put a patient support system together that will both educate and give them a forum for their voices to be heard. We'll introduce that a little later this year. Progress. While I was setting up n-Lorem, we had the opportunity to help 2 investigators treat 14 patients. That experience was really valuable, particularly because it anticipated the guidance from the FDA By far the most informative are Neil Schneider's 11 FUS ALS patients. FUS ALS is arguably the most aggressive form of ALS. And typically, I think of average lifespan after symptom onset is 16 months. This paper, if you haven't seen it, is worth reading. Very interesting. A pair of identical twins, one of whom developed symptoms of a FUS ALS when she was 16 and died promptly. And then her sister, identical twin, didn't become symptomatic until 25. This is one of the urgent cases we had. She was in hospice on a ventilator. So we really didn't think we'd be able to get the drug to her, but she was treated and she did get stronger. She did get out of the hospital, and she's really recently succumbed to the disease. Probably more informatives are the other 11 patients that Neil has treated, all of whom are getting stronger and appear to be doing far better. And our favorite is Anna, who's a teenager in Germany, who was in pretty desperate straits. She had an obstruction from food and ended up getting pneumonia, aspiration pneumonia. And as Neil says, no one with FUS ALS ever gets better except Anna and the other patients. She's now able to walk unaided and is able to rise unaided, breathing on her own and going to school, which is hard to argue that could be just a part of the natural progression of the disease. So Neil's putting a paper together describing all this, and I recommend it to you. What about systems? I mentioned the system, the industrialized system is vital. Obviously, a great deal of work has to happen before we can do anything, and that's done in tertiary care, personalized medicine centers. The patient must be fully genotypically characterized and phenotypically characterized as well. And we need to know what the gene function is and that the mutation that's been identified is causal. All that comes. What happens is a research physician, it does take that, and patient or parent complete an application on the n-Lorem website. We then triage that, do all the literature work, understand the character and the prevalence of the disease. And then that patient is presented to a committee I put together called the Access to Treatment Committee, which is made up of all the expertises that we need to make these incredibly complex risk-benefit judgments. These are sick patients with multi-organ manifestations, and many have developmental overlays. It's tough stuff. And then with their advice, then I make the final decision about who's the right kind of person to treat with ASOs. Once we make that decision, then we work with the physician to define primary, secondary, and exploratory treatment goals and clinical measures that we'll use. During the 15 months or so that is currently taking, we hope to get that down, to get the ASO to the patient. We ask that they perform very careful natural history, and then we compare that to on treatment in the first year. Yes, it is true there are n-of-1 trials designs, but they're all crossovers, and that won't work here because these patients are too sick and the duration of effective ASOs is far too long. So we think this is the best solution to attempting to evaluate the performance. As I said, demand has exceeded my expectations by maybe 30-fold. We're now up to almost 150 applications. I think we've approved for treatment about 60. We just had our first pre-IND meeting with the cardiorenal division, and that went extremely well. And that's a patient with renal amyloidosis. That's a very interesting case. And so the guidance is working. And in response to the demand, of course, I've had to grow the place a lot more rapidly than I expected. And I'm very pleased with the team that we have been able to assemble. This is a galvanic idea, and people want to be a part of it. And I'm thrilled that the quality systems that we've put in place appear to be working well. I mentioned that we have a lot of collaborators and donors and partners. This is the list today. I expect by the end of the year not to be able to fit this on a single slide. What's compelling to me is a number of biotech companies, including Ultragenics, and every single vendor and provider in the industry has joined, and they're giving us tremendous discounts and also cash donations, and that means that our cost per patient at steady state I project to be around $600,000 to $800,000 a patient, all in, not millions. And of course, that number makes it feasible. And these collaborations have allowed us to reduce cost per patient by 40% in the first couple of years. We have more to do. So we are publishing. Here are the papers so far from n-Lorem. Should you be interested, you can learn more about what we're doing and how we're doing it and why we're doing it in some of these papers. And so I'll conclude by saying the nanorare patient is the most isolated, desperate, and needy and underserved patient population I've encountered. I think we're off to a great start. I think by any measure, I'm pleased by where we are, but we know we're just beginning. And the next task for us is to demonstrate a nonprofit model is actually sustainable. Given the progress, I'm optimistic it will do that, but we're going to need all help we can get. And so anybody interested, just give me a call. I'll put you to work. Thank you.